Assalamu alaikum and welcome to Biology School. A microscopy is a device that is used to see the small objects as like cells. To know the cells and to know the biology, it is important to know about the microscopy. So in this lecture, we will discuss about in short brief on microscopy. In this lecture, we will discuss about microscopy, different parameters of microscopy, and finally, we will discuss about two commonly used microscopy, those are light microscopy and electron microscope. But first, we will just know about what is microscope. A microscope is a scientific device that is used for magnifying objects too small to be seen by naked eye, producing an image in which the object appears larger. Actually, microscopy comes from the Greek word micros, that means small, and scopion, that means to examine. The parameters that is commonly used in microscopy, and it is urgent to know in details about what kinds of parameters are used in microscope. The one is magnification, second one is resolution, and third is contrast. Magnification is the ability of a microscope to produce an image of an object at a scale larger than its actual size. Mainly, magnification is the ratio of an object's image size to its real size. We can say that it will be the magnification is equal to dimension of the feature in the image divided by true dimension of the feature on the object. Light microscopes can magnify effective to about 1000 times the actual size of the specimen at greater magnification. Additional details cannot be seen clearly. Actually, when we see any specimen under the microscope, so how many times it will be clear to us, that actually expressed in the case of magnification. In the resolution, the term resolution is used in microscope to express the ability of a microscope to distinguish details of a sample. Resolution is a measure of the clarity of the image. It is the minimum distance two points can be separated and still be distinguished as separated points. For example, what appears to the unaided eye on the star in the sky may be resolved as twin stars with a telescope, which has a higher resolving ability than the eye. Similarly, using standard technique, the light microscope cannot resolve detail finer than about zero point 0.2 micrometer regardless of the magnification. Contrast is defined as the difference in light intensity between the image and the adjacent background related to the overall background intensity. It describes the brightness between the light and dark areas of an image. Staining or leveling cell components are the method for enhancing contrast to stand out visually, that is practiced in the laboratory to find out the contrast of the specimen to observe it clearly. The microscopes used in biological research, the mainly light microscope and electron microscope. Light microscope is any microscope that uses visible light to illuminate an image specimen. Light microscope includes white light composed of all wavelengths, as well as the light of a specific wavelength used in fluorescent microscopy. Light microscope is any microscope that uses visible light to illuminate and image a specimen. Usually, when people refer to light microscopy, they refer to non-fluorescent microscopy, even though fluorescent microscopy does, of course, use light. Modern light microscopes, which operate with visible light, use two magnifying lenses to achieve very high magnification and clarity. The first lens focuses the image of the object on the second lens, which magnifies it again and focuses it on the back of the eye. Microscopes that magnify in stages using several lenses are called compound microscopes. So there are various types of light microscopes that are bright field light microscope, phase contrast light microscope, dark field light microscope, and fluorescence light microscope. An electron microscope is a microscope that uses a beam of accelerated electrons as a source of illumination. It is a special type of microscope having a high resolution of image, able to magnify objects in nanometers, which are formed by controlled use of electrons in a vacuum captured on a phosphorescent screen. Ernest Rusko, a German engineer and academic professor, built the first electron microscope in 1931, and the same principle behind his prototypes still govern modern electron microscope. Rather than focusing light, the electron microscope focuses a beam of electrons through the specimen or onto its surface. Resolution is inversely related to the wavelength of the light a microscope uses for imaging, and electron beams have much shorter wavelengths than visible light. Now, in short, about the working principle of electron microscope. The electron gun generates electrons. Two set of condenser lenses focuses the electron beam on the specimen and them into a thin, tight beam. To move electrons down the column, an accelerating voltage is applied between the tungsten filament and anode. The accelerating voltage mostly between 100 kV to 1000 kV. The specimen to be examined is made extremely thin, at least 200 times thinner than those used in the optical microscope. Ultra thin section of 2200 nanometer are cut, which is already placed on the specimen holder. The electronic beam passes through the specimen, and electrons are scattered depending upon the thickness or refractive index of different parts of the specimen. The denser regions in the specimen scattered more electrons and therefore appear darker in the image, since fewer electrons strike that area of the screen. In contrast, transparent regions are brighter. Then the electron beam coming out of the specimen passes to the objective lens, which has high power and forms the intermediate magnified images. The ocular lenses then produce the final further magnified image. There are two types of mainly electron microscope, the scanning electron microscope and the transmission electron microscope. The scanning electron microscope is especially useful for detailed study of the topography of a specimen. The electron beam scans the surface of the sample, usually coated with a thin film of gold. The beam excites electrons on the surface, and these secondary electrons are detected by a device that translates the pattern of electrons into an electronic signal sent to a video screen. The result is an image of the specimen's surface that appears three-dimensional. And transmission electron microscope. The transmission electron microscope is used to study the internal structure of the cells. The transmission electron microscope aims an electron beam through a very thin section of the specimen, much as a light microscope aims light through a sample on a site. For the transmission electron microscope, the specimen has been stained with atoms of heavy metals which attach to certain cellular structure, thus enhancing the electron density of some parts of the cell more than others. The electrons passing through the specimen are scattered more in the danger region, so fewer are transmitted. The images displays the pattern of transmitted electrons. Instead of using glass lens, both the scanning electron microscope and transmission electron microscope use electromagnets as lenses to bend the paths of the electrons, ultimately focusing the image onto a monitor for viewing. And by this we can clearly see the structure of cell. And this is the short discussion about the microscopy that is used to study in the cell. For this lecture, we take the support of few sources, and that is the Campbell Biology, Raven Biology, Khan Academy, Science Direct Journal, Leisa Microsystems, and Byju's, and biologyschool.net. Thank you very much, and bye bye, and see you for upcoming lecture.